Your genes are the molecules inside your cells that tell your body how to make proteins. That is, they explain what amino acids the protein is built out of and in what order. Let's take a look at how this information gets from your genes to the final protein through a series of processes called transcription and translation. Let's take a closer look at a gene. A gene consists of three main parts: the promoter, the coding segments, and the stop codon. The DNA never leaves the nucleus. However, proteins are built in ribosomes outside the nucleus. So how does this information get from here to here? The answer is messenger RNA, which acts like a mailman, delivering information from the nucleus to the cytosol. The key enzyme involved here is RNA polymerase, because it builds the RNA polymer from monomers called nucleotides. RNA polymerase floats around the nucleus, and when it finds a promoter on the DNA, it binds to it and begins moving down the gene. As it moves down the DNA, it unzips it into two strands, called the coding strand and the template strand. RNA polymerase hugs the template strand, and as it moves down, it begins building an mRNA molecule, which is complementary to the DNA. If it sees a T, it adds an A. If it sees a G, it adds a C. If it sees an A, it adds a U, and so on. This continues all the way to the stop codon, a series of 3 bases which tells the enzyme that it's reached the end of the gene. When it sees this, RNA polymerase pops off and releases its strand of mRNA, and then moves on to transcribe another gene. This strand of RNA might look ready to leave the nucleus, but it's not quite there yet. This is only pre-mRNA, or precursor mRNA, and there are a few things we need to change first before we can move on to translation. The first is a process called splicing. In every gene, and hence in every pre-mRNA, there is some redundant, useless information, sequences of base pairs which don't contribute to the protein. These segments are called introns, while the useful segments are called exons. An easy way to remember this is just to remember that exons get expressed in the protein. Molecules called spliceosomes act like scissors on the RNA, cutting out the introns and recycling their materials. The leftover exons are then glued back together by another enzyme called RNA ligase. Sometimes, in order to create a number of different proteins from a single gene, spliceosomes will cut the same mRNA molecule differently at different times, removing some exons one time and then removing different ones the next. This is called alternate splicing, and it's the reason why the human body can create over 100,000 proteins from just 20,000 genes. For example, this is how your body can create a different antibody for just about every possible virus or bacterium from just a handful of genes. The second step is to add a poly-A tail, which is a sequence of about 200 adenine bases which are added to the 3' end of the molecule. Imagine somebody was typing out the base sequence for the RNA and then fell asleep on the A key. That's essentially what the poly-A tail is. This tail helps to prevent the RNA from degrading due to other enzymes present in the cytosol. The final third step is to add a 5' cap, which is really just an altered guanine nucleotide. Again, this helps prevent the RNA from degrading due to the presence of other enzymes in the cytosol. So now we've spliced out our introns, we've added our poly-A tail and our 5' cap. We're ready to slip through the nuclear pores and head out into the cytoplasm. Just like words can be translated from one language to another, information from mRNA can also be translated from nucleotides to amino acids. In this case, the translating is done by the ribosome. The ribosome is made up of proteins and ribosomal RNA, or rRNA for short. It consists of 2 subunits, the large upper subunit and the small lower subunit. The mRNA is wedged between these 2 subunits and feeds through from one end to the other, a bit like a ticket reader. mRNA is read by the ribosome, one codon at a time. A codon is a sequence of 3 bases on RNA that code for one of the 20 amino acids. For example, the codon AAA will tell the ribosome to add the amino acid lysine. AGA tells it to add arginine. UAC tells it to add tyrosine, and so on. Here we introduce a new molecule, transfer RNA, or tRNA for short. As its name suggests, tRNA transfers the needed amino acid to the ribosome. On one end of the tRNA is the anticodon, which is complementary to the codon, and attached to the other end is the amino acid that corresponds to that codon. The tRNA with the correct anticodon will bind to the mRNA at the rightmost site of the ribosome. We call this the A site. Then it shifts over to the second site, called the P site, allowing a second tRNA to bind with the A site. With the two tRNA molecules firmly held in place, a peptide bond can form between the two amino acids, starting our polypeptide chain. The first tRNA detaches from its amino acid and shifts over to the final E or exit site, where it detaches from the ribosome and leaves to find a new amino acid. Now, the next tRNA for the next codon can move in, allowing the next peptide bond to form. This process repeats over and over, continuously reading codon after codon and adding amino acid after amino acid to a growing polypeptide chain, until it reaches the stop codon. There is no tRNA for this codon, so the entire ribosome falls off and the polypeptide chain is released. It can now head to the rough ER or to the Golgi body for processing into the final protein.